Okay, today we're going to be talking about testing. Okay, I think we got it. Today we're going to be talking about proteins, specifically a certain type of protein, which we call enzymes. And as I mentioned in class and how we've been talking about, proteins have many functions. And one of the functions is that it can either speed up or slow down chemical reactions, in that it breaks chemicals apart. Okay, so that's what we'll be talking about. So before we start, we're going to have to get some terms down, and more or less, chemical reactions don't always occur spontaneously. Sometimes they need a little help to happen, and this help that they require to have them happen is called activation energy. So let's talk about the lab that we are going to be doing in class. In class, we're going to be working with hydrogen peroxide, which has a chemical formula of H2O2. So we'll call that hydrogen peroxide. Okay, and this is a chemical that has more or less two hydrogens and two oxygens in the center, and it's all connected together. In the lab, or actually just in everyday life, hydrogen peroxide will naturally break down into H2O, which we know is water, and oxygen. And I hope all of you are looking at your notes and definitely writing this down. So make sure that you have that included. So, so hydrogen peroxide breaks down to water and oxygen, and it often takes about 24 hours actually to do this. So it takes a long time for certain standards. However, however, we can get it going faster. We can do it faster with the help of an enzyme. And what it does is the enzyme will change its activation energy. That means that it does not have to take so long, or it doesn't need to put in so much energy to break it apart, because the enzyme actually helps breaking it apart. And we're going to look at exactly what that looks like. So let's move on to the next slide. So enzymes are able to either to speed things up a little bit. So reading what I have written here, some chemical reactions make life possible that are too slow, or have activation energies that are too high to make them practical for living tissue. Again, these enzymes are what we call catalysts. And these are enzymes. And what catalysts do is they lower the activation energy. Activation. Do we have it? Activation energy. Okay? So in other words, it makes it easier for the chemicals to come apart. And enzymes are considered these catalysts. So enzymes can make it easier for chemicals to come apart. And I know in a second there's going to be some writing on top of that, but let's see. There we go. And enzymes, more or less, what they do is they speed up chemical reactions that take place in the cell. And they do that by lowering the activation energy. All right, and it would be the example of two students, and we did this in class. And they're holding hands. These are my chemicals, or the substrate. And with the help of an enzyme, they are broken apart. And that enzyme is adding energy in here so that you can break them apart easier. So this graph is a classic graph that you're going to be seeing in class, and you're going to have to know it. So here I have my hydrogen peroxide. We call it a reactant. And in the end, I'm going to have my product, which I call water and oxygen. And you don't need to worry about reactants or products. I will always tell you which ones they are. And normally, without the help of an enzyme, it needs this much energy pumped into it. So it's a lot of energy pumped into it in order for these to break apart. And here's where they start breaking apart. Okay? And then you, at that point, have the graph going down because it's broken apart. But take a look, this one here. With the help of an enzyme, look at, you need to have a lot less energy. All right? This energy required is only from here to here. Unlike this one, which requires energy from here to here. So with an enzyme, we don't need so much energy. Don't need so much energy. And so it breaks it down easier and faster. So what does that actually look like when we are looking at the enzyme? And let's identify some of the terms, okay? So first of all, I have here my enzyme. There it is. This example is hexokinase. Don't worry about the name. Just always know that enzymes always end with an ASE. And you can see the strange shape. And somewhere in that shape, I know you're going to have spirals, you're going to have beta pleats. They've all been wrapped in together. And that enzyme, in its someplace, has something that's called an active site. And this place, this active site, is where you have your chemicals coming in. So here, I have two chemicals. I have glucose, that's substrate 1. We can also call it substrate. Okay, so there's glucose, and I have substrate 2, which is ATP. You do not need to worry about what they are. Just know that you have two different substrates, and they will float into the activation site, or the active site of your enzyme. So they're moving in there, and when they move in there, the enzyme starts making connections happen. So take a look here. You have 4 little blue dots, and the enzyme causes those, 3 little blue dots. You have then 2 blue dots still attached to this here, but this time around you have 1 blue dot attached to the hexagon, so that you now have a chemical reaction that happens, something that changed with the help of an enzyme. This glucose looks different now. It has a blue circle on it where it started off having no circle on it, and this here has 1 less circle. The enzyme let that reaction, or made that happen, in its active site. And that's where I am going to end the film, and I will also include a video that you can find in your links page. Be sure to answer the questions on your quiz as well.